Hi, and welcome to this presentation about glioblastoma created by myself, Andrea Olsen, and my mentor, Anastasia Schneiderman. So we have created this investigation in aim of updating the current glioblastoma example study on the Pandomics website. We'll begin with a general overview of what glioblastoma is, so its molecular mechanisms, traits, and treatment. Glioblastoma, also known as grade 4 glioma, is the malignant cancer of astrocyte cells. Astrocyte cells are a type of glial cells that are widely known as support cells of neurons. However, recently, researchers have started theorizing about other qualities that they might have. Glioblastoma can be located anywhere in the brain, and normally it occurs in people between the age of 45 and 70 years, and it's not common in children. The most widespread symptoms of this tumor are listed on the slide, and you can see that it's not only physical, but also cognitive impairments like memory loss and language deficit, as brain structures important for these tasks are also damaged. Treatments include surgery, chemotherapy, radiation therapy, sometimes immunotherapy, and alternating electric fields therapy. And we'll get into some examples quite soon. Unfortunately, this is a very severe cancer, so even patients that undergo a powerful combination of various therapies live on average 14.5 months post-diagnosis. Here are some pathways and genes highly associated with glioblastoma. So the most famous ones are probably IDH1 and IDH2 mutations, and in fact, their presence defines whether glioblastoma is of primary or secondary cause. Primary glioblastoma forms directly from glial progenitor cells, and it compromises 90% of cases and has the IDH wild type. Secondary glioblastoma develops from lower grade gliomas and carries mutations in IDH. And some other frequent mutations include alterations in NF1, PTEN, and MDM2. The study of epigenetics in glioblastoma multiforme has become famous lately. Powerful input from this field has come during the last several years from the discovery of frequent methylation of the promoter gene MGMT, and found in about 20% of glioblastomas. And this promoter performs DNA repair and can fix the DNA damage from alkylating chemotherapy drugs, such as temozolomide. A well-known pathway in glioblastoma is the PI3K ACT pathway. It causes the upregulation of many different genes, and the ACT gene has an important role in tumor proliferation and radiosensitivity and can be inhibited by drugs to prevent ACT translocation to the cell membrane. EGFR, a gene that's also upregulated by this pathway, has shown to be a major biomarker of glioblastoma, and thus inhibiting this pathway has also decreased EGFR expression. The RAS-MAP-ERK pathway is another one that's associated with this cancer, and a study showed that inhibiting it actually reduced cell proliferation significantly, which stopped the spread of malignant glioma. Next, a study demonstrated that the mTOR expression was significantly higher in wild-type IDH1 primary glioblastoma when compared to healthy tissue. This pathway is known for promoting cell growth by phosphorylating two downstream targets that drive protein synthesis, and also for contributing to protein synthesis by activating the transcription intermediary factor 1 alpha. And finally, the WNT and B-catenin pathway. It is known as an important regulator of intracellular interactions, sulfate decision, and migration. The existing literature establishes that WNT plays a significant role in maintaining stemness in normal stem cells, thereby allowing repair and regeneration. However, a deregulated WNT pathway may result in an onset of cancer stem cells. So regarding glioblastoma, it's still difficult to judge just how important this pathway is due to cross-talking pathways and a large number of ligands and receptors involved. Nevertheless, with new incoming evidence, this pathway should not be disregarded. And now please shift your attention to the fourth pathway. Many drugs, such as bevacizumab, have targeted the VEGF gene lately to treat glioblastoma. Analogous to our study, there was another investigation made by Tang and his team in 2018, and this image shows the datasets that they chose to use. We compared our own results with gathered datasets in Gene Expression Omnibus, and found that we had two matching datasets. The reason why we just have two datasets that are the same is firstly because this study was conducted a few years ago, and since then, new and more recent publications of glioblastoma have been produced. And secondly, we have a strict and specific requirement for the data sets that we choose. For example, all of our data sets had to have at least 4 control patients so that we could conduct comparisons of gene expression on pandomics inside each separate dataset. And we also chose datasets studying specifically glioblastoma, while these researchers in the study included other brain tumors as well. In addition, the ones that we chose had to be published not earlier than in the past 10 years. And finally, all samples taken in these datasets must have been from brain tissue. Such requirements make our investigation highly reliable. So the mentioned study compiled a list of genes that were up and down regulated in glioblastoma patients. In this table, we can see the roles they play in this cancer, which could indicate certain ways in which we could target these genes in the future. After our own meta-analysis, we compared the results to their table. Here you will see the most popular treatments of glioblastoma multiforme. Unfortunately, we haven't found a dataset which gives us both pre- and post-treatment results, which is why we cannot show specific expression of genes in treated and non-treated patients. However, we still get some good insight from the established literature. So from chemotherapy, the most well-known one is temozolomide, also known as Temodar, and this is an alkylating agent which destroys DNA in glioblastoma cells. And from the attached Kaplan-Meier graph, you can see that the overall survival increased for those taking the drug. And although it seems that just by a little, the years of added life are still significant. From targeted drug therapy, there is bevacizumab, also known as Avastin, which is designed to block VEGF. Blocking VEGF may prevent the growth of new blood vessels, including normal blood vessels and blood vessels that feed tumors. And regarding immunotherapy, erinda is a peptide-based vaccine targeted against EGFR surface antigens. And in the image on the right, you can see its mechanism of action. Unfortunately, there aren't many good drugs against glioblastoma yet, so we are highly motivated in finding new druggable targets so that the new treatment methods can be established.